Okay. So first of all, we really want to thank all of you and your families for being a part of our research program. This has not been an easy year for anyone, and we certainly recognize that. And we do thank you for your continued engagement with the study and for the support that you have shown us and your enthusiasm about coming back when the time is right for each one of you. And many of you have already started coming back to the study. And really, there would be no study without you. So we want to thank all of you for all that you have contributed to this work. Additionally, we'd like to thank the Longevity Genes Project, LonGenity, and Lifelong Study teams. These include Debbie Truck, who is our nurse practitioner, Tina Gao, and Dr. Galia Lodish, who has joined our team recently. You all know Wanda Guzman very well, and she's really the engine behind LonGenity. And I think without her, we wouldn't be able to run a study. And she has adapted during these times and has made the study adaptable to meet the challenges of these times. Maria Mangan, who many of you know, Geeta Perswat, Kathy Rosales, and Dr. Erica Weiss. So I want to take you through a brief history of our longevity studies here at Einstein. It's a little bit of a timeline, and to also put things in perspective about where we have been, where we are, and where we're going. So back in 1998, Dr. Nir Barzilai, who's the director of the Aging Institute at Einstein, had an idea to begin the Longevity Genes Project, and that is the first study that began. And that study focused on centenarians and their families, and he was interested, oh, I apologize for that, and he was interested in centenarians because they don't only live long lives, but they also live healthy lives. So they really epitomized healthy longevity, which is what we're all after. We want to live not only long lives, but productive lives. And that was really the start of the longevity program. In 2008, there was a phase 2 of the program, the LonGenity study, and that is the study that you're most familiar with. This is a study that enrolled individuals who are 65 and older, those who have a parent with exceptional longevity, and those who do not. And at this point, I'm proud to say that we have about 1,200 individuals involved, many of whom have been with us from day one, and we're still continuing to enroll people in the study. And you'll hear about some of the new initiatives that we have as we're always trying to stay on the cutting edge of science and implement better investigative tools so we can learn more about longevity. Now, many of you may have had parents who were participants in the Longevity Genes Project, and some of you may have participated in that first phase as well. And we do have some LonGenity participants who are now reaching the ripe age of 95 and are also enrolling in the Longevity Genes Project. And a decade later, in 2018, we have conceived a phase 3 of the study, and that is the lifelong study. And this study includes the grandchildren of our LonGenity participants and maybe the great-grandchildren of the centenarians and the children of the LonGenity participants. So this study looks at people who are younger at the age group of 20 to 59. And it is important for us to study these younger people because we know that aging doesn't miraculously start at the age of 65. We know that aging is a continuum. And in order for us to have a good understanding of the aging process, we need to first be able to compare older people to younger people to see what are the changes that occur with aging, but also to see the differences in younger people and how they age. And at this time, we have enrolled about 200 people in the lifelong study, and we're still looking to enroll others. So I would encourage you, if you have children or grandchildren who are interested in participating, please reach out to us. This is not an onerous study. It's a 30-minute visit, or we also go out to visit the individuals in their homes. It's not as involved as LonGenity, but it gives us valuable information about health history, and we also collect a blood sample for DNA and other parameters. So if your children or grandchildren would like to participate, please reach out to Wanda and she'll put you in touch with Kathy Rosales, who's the coordinator for that project. So now I really want to take a few minutes to share some of the new initiatives that we have in LonGenity, some of which you're already familiar with. So COVID is obviously very much in everybody's mind, and we know that older individuals in particular have been most affected by COVID. We know that aging is a major risk factor for mortality and morbidity in the setting of COVID, and Dr. Barzilai will talk more about that today. But we recognize that we have an opportunity and a responsibility to study COVID in the setting of our LonGenity study. And so this is a new study that we initiated that is currently funded by the NIH and the National Institute on Aging. And it's led by Dr. Helena Blumen, Dr. Erica Weiss, and myself. And what we aim to understand is the impact of COVID-19 infection on emotional and cognitive health in older adults, and the impact of social isolation on emotional and cognitive health. And by social isolation, we mean the social distancing requirements that have been implemented in the setting of COVID that have required that older adults isolate themselves to some degree in their homes. And we know that many have not seen their grandchildren or children for a long time in order to maintain safety. And we wanted to study the impact on that on individuals' emotional and cognitive health. What is involved in this new initiative? Well, we do COVID-19 antibody testing to evaluate for prior COVID infection. This is a part of routine blood testing. We also have implemented some neurocognitive evaluations via a Zoom platform, and a subgroup of individuals are eligible for this part of the study. We have added questionnaires about social support systems that are specific for this very unique time period in our history, and participation routine LonGenity visits. So by collecting this additional information, as well as participating in your usual annual LonGenity visits, we'll be able to understand how the COVID-19 pandemic through infection or through just social isolation has impacted aging and cognition in older adults. Another initiative, which some of you may already be familiar with, is focused on Alzheimer's disease, dementia, and brain aging. This is an initiative that is also sponsored by NIH and NIA, and there are actually 4 major grants that we have from the NIH that are focused on this question, totaling about $10 million. And these are research projects that are led by Dr. Barzilai, Dr. Blumen, Dr. Alexic, and myself. And these have several aims. One is to understand the resilience to Alzheimer's disease and dementia. We know that centenarians and the children of centenarians are more resilient to developing dementia. They develop it later in life, or they don't develop it at all, despite advanced age, even though age is a major risk factor for dementia and Alzheimer's disease. And we want to understand what is it about centenarians that protects them from cognitive decline. Another aim is to understand the role of social networks in GAIT, in brain and cognitive aging, and Dr. Blumen will speak more about that today. and to understand the role of hypothalamus in aging, which is a part of the brain, which is a master regulator of many hormones and metabolic processes. And Dr. Alexic will touch on that today as well. What is involved in this component of the study? Well, we conduct brain magnetic resonance imaging, or MRI imaging, for eligible and interested participants. This provides us with information on brain structure, and Dr. Blumen has a protocol where individuals are asked to imagine walking, which she will talk more about today, and that provides information on brain function. We also incorporate a social support questionnaire to understand how our social support systems may actually impact the structure in our brain. And participation routine visits, because all the information that we collect from the annual visits is also used for better understanding of dementia, Alzheimer's, and brain aging. And a third initiative, again, which some of you may already be familiar with, is the coronary CT study. That is led by Anna Bortnick, and it's also supported by NIH. And the aim here is to understand resilience to cardiovascular disease. So what factors protect people as they age from developing cardiac disease or cardiac calcifications or valvular disease of the heart. And this involves a CAT scan of the heart, looking specifically for calcifications in the arteries of the heart and in the valves of the heart. There's a blood draw, and again, participation routine visits. So by coming for your annual LonGenity visits, you are contributing to all of these specific initiatives, but you can also make a choice to do additional studies like the coronary CT, the MRI, or participate in the COVID protocol. Now, if you choose not to participate in any of these sub-studies, you're still welcome to participate in LonGenity, and we're learning a lot from those visits as well. So we have a really exciting program for you today. We will begin with Dr. Nir Barzilai, who's the professor of medicine and genetics at Einstein and the director of Einstein Institute for Aging Research. He will give you an overview on aging and longevity and what we can do about those processes, because aging is not an inevitable process where things can be done to intervene. Then we'll hear from Dr. Helena Blumen, who is an assistant professor of geriatrics and neurology at Einstein. And finally, we'll hear from Dr. Sandra Alexic, who's an assistant professor in endocrinology and geriatrics, who will talk about brain regulation of endocrine and metabolic systems. And we'll conclude with a question and answer session after the talks. So the format for today will be as follows. We will first hear the talks from each of the speakers. If you have any questions for the speakers, please type them into the chat feature that should be on the bottom of your screen. If you don't see it, you may just need to hover with your mouse. And there's a Q&A icon there. And if you click on it, you can type in your questions. And at the conclusion of all the three talks, I will moderate an question and answer session with our speakers where they will be answering your questions. And with that, we will begin with Dr. Nir Barzilai.
Thank you so much. I have to thank somebody that hasn't been thanked before, and that's Sophia herself. Sophia Millman is an insightful and clever investigator, multitasking, and has set us to the next stage. And it's just an honor for me to be her collaborator and even more important friend, as well as with Helena and Sandra, and you'll hear them next. So before sharing the slides, I'll tell you the only thing that is really important in what I'm going to say. And that is that you know that there is a biology of aging because you know who's old and who's young. But we figured out, and in part in Einstein and in part in this study, that aging is actually quite flexible and we can change it. And I'll give you the rationale of why we should change it. And now I'll share my slides and tell you that what my study in historically in the 1998 has begun with this family in line, and this is our poster child and family that has participated in longevity gene study and in LonGenity, and that's the Khan family. And that's their picture taken between 1910 and 1920 when they were born, or 10 years later or so. And the important thing about this family is that all of them lived past the age of 102. The older sister, Helen, died at 110. And then the others at 106 and 108. How is it possible that four siblings that are living in New York slow their aging so much and live so long? And not only live long, but when I met Helen the first time, she was smoking. She was out of her apartment in New York smoking. And I said, Helen, nobody told you to stop smoking? And she said, you know, all the four doctors that told me to stop smoking, they died. In other words, those people not only lived healthier and live longer, but also were resistant to some of the things that we know should age them a lot. And this is the beginning of our study. And now we have a lot of them. Okay, we have almost 3,000 people in our study, out of which 750 approximately are centenarians, and the rest are either offspring of centenarians or people without a longevity in their family. And to learn more about it, you can look at my book that came last year, and the title of the book is Age Later. And I just want to read to you what I wrote in the acknowledgement, because the first people in the acknowledgement were you. So I want to make sure that you know how we appreciate that. And I'm saying this book and my work wouldn't have been possible without, okay? And talking about you volunteers, these volunteers agreed to be interviewed and allowed us to take blood samples and conduct variety of tests, including MRI of the brain and CT scan of the coronary arteries. Many of them participated in the study throughout the years, showing up rain or shine, and are inspiring to all of us. It is a beautiful partnership, and I wish them all extended health span. Okay, so that's about you. And your partnership is evaluated, and that's why we are doing those events, and that's why we're welcoming questions and other suggestions. So when we came into the field, what was clear to us, it's what intuitively is clear to you, that as we age, our chances of dying from any one of those diseases here is going up. But please notice that here the y-axis is what we called a log axis. In other words, if you take heart, you go from death of 5 out of 100,000 when you're in your 20, when you're in your teens, to 5,000 when you're over the age of 85. And this is a greater risk than, for example, cholesterol, which is kind of 3 to 5-fold, or smoking, which is 10-fold. There's no greater risk of dying from aging from those diseases than aging itself. It's similar with cancer and with diabetes and Alzheimer. And the hypothesis, the leading hypothesis, is that this aging, this biology, is what's driving the diseases. We made a kind of a weird a deal with the devil, you know. When our life expectancy was 35, we begged to live longer. And so the devil said, fine, and gave us water and sewers and immunization and antibiotics and surgery and everything. And what did it get us? To much more awful diseases like this cancer and Alzheimer and all those. And it's clear to us that we can stop it, but in order to stop it, we have to stop aging itself. There's nothing that exemplified it more than the COVID. And this is a slide that has panel from China and Italy and the US and the EU. And it doesn't matter what those panels show, they show the same thing in just different ways. But if you look at China, if you're over the age of 80, your chances of dying is 180 times more than when you're in your 20s. This virus that has no eyes just knew how to pick on the older adults. Now, we have established this field that is called geroscience. You know, it used to be called anti-aging or something like that, but this has been populated by charlatans and people who are selling snake oils. We're actually scientists. And from your perspective, what is geroscience is to understand those changes that we see regardless of the disease. The sleep pattern, the fact that we get more infection, the fact that our blood pressure is crazy, our skin is dry, we're cold, our balance is not good, we're changing weight, our strength is declined, our mobility, our cognition is declined. But in fact, when you turn it into scientific terms, we're talking about hallmarks of aging. And actually at Einstein, we have several centers that are investigating basically all those hallmarks of aging. And I'm putting in those hallmarks of aging not to go over them, but to point really two things. First of all, you see that the immune dysfunction is one of them. That's why with aging, we get more COVID. Not only that, there's inflammation, and inflammation is really the process that kills people like in the 5th day. That's why they get into trouble. But the nice thing about your seeing is the interaction, the lines in between, because for those hallmarks, and to be a hallmark, you have to show that it changes in aging and that if you fix it, you get health span or lifespan extension. That's how you become a hallmark. And those are knobs, okay? And if you turn the knobs on, you get effect not only on one, but on the other hallmarks as well. So we're trying to do all of it, but it's enough to do some of them to gain an unbelievable effect. And through the years, we studied longevity, and we basically found two things. That first of all, healthy lifespan, and we're talking about health. That's the most important thing. I'm always kidding that you know, health and longevity will go together, but longevity is the side effect. You know, maybe you cannot afford to live long, but health span and lifespan has been extended. We've done tons of studies about that. And not only that, but relevant drugs are even in human use. So when it comes to the centenarian, the first thing that we really cared about was, so just a minute, are they getting sick when all of us getting sick? And all they do is live sick for longer? Or are they living longer and living healthier? And this is really the results. What we looked at is disease-free survivors. So everybody doesn't have disease at the beginning. And then people are accumulating the disease. So people after age of 60, fewer and fewer are disease-free. You see that at age 80, only like 10% are disease-free. And you see that our centenarians have been disease-free for longer, for like 20, 30 years longer. And even at age 100, like almost 30% of them, then they don't have a disease. So their aging is slow, okay? Their aging is slow. A lot of your aging is slow, guys, out there. And this is really the showing that we have the capacity to live healthier longer, considering we die before average age of 80, we have much, much better capacity. But you know what? This is not the most exciting thing. The most exciting thing is they have what we call contraction of morbidity. They're sick for just very short time at the end of their lives, not like us that accumulate diseases for years. They just, some of them, those 30% don't have a disease. They just don't wake up one day. And this is really translated to what we call longevity dividend. You know, the cost of not doing it is incredible. The cost of not changing aging is incredible, because if you live longer and healthier, that's a huge saving. In fact, the CDC that we all heard a lot about for years, this is data from 93, showed that the medical cost in the last 2 years of life of somebody who dies at 100 is about third than those who dies at 70. So they live happy, they live healthy, okay, they die, and everybody saves a lot from that. We can invest in science again and in green technology and all that. By the way, we have discovered several longevity genes and have developed, and several drugs have been developed based on our finding. I just want to end by giving you a glimpse on the future, and there's more glimpse in the future on my book that you helped me write. But think of it this way: what you see here is a fertilized egg. You can so a new baby is formed, okay, and you can get a sperm of a 70-year-old man and an egg of a 50-year-old woman. You know, it's been done before. And you can fertilize the egg. But the important thing is, when this baby is born, he doesn't remember the age of the parents. He starts anew. And this mechanism is something that is already in investigation, how we can erase aging. I mean, we figured it out in this example, but could we think of a future where we take a 20-year-old and every month give a certain treatment and erase the aging, and we stop going old? Is this a possibility? We certainly think that this is a possibility, and we're looking at that. I also would say just that that this is not only aging. You know, people who are surviving chemotherapy and radiation, which are things that ages you, they need help. And people with HIV age 10 years, they get diseases 10 years earlier than their friends. And people who are disabled, who are in wheelchair and maybe eat more and cannot move, they need help. And talking about the future, if we want to go to Mars, we have to stop aging, because we cannot go there now without getting cancer or cardiovascular disease, and we'll never get back. So we're on a bigger mission than just helping us. Now I'm formally an elderly too, but this is something that the world needs. I'm thanking you again. I'm thanking all this wonderful crew, and enjoy the rest of the lectures. Thanks so much.
Thank you, Nir. And I just want to remind everyone to please type your questions in the question and answer box. And now it is my pleasure to present to you Dr. Helena Blumen. Dr. Blumen is a PhD psychologist. She received her PhD in experimental and cognitive psychology, and she's studying the control of gait and cognition by the brain. She is an assistant professor of geriatrics and neurology at Einstein, soon to be an associate professor. She's received numerous awards, one of which is an Outstanding Junior Research Award for her work on control of gait by the brain, and she will share that work with you today. Helena?
Thank you, Sophia, for Dr. Millman. Can everybody see my slides for those of you who are co-hosts?
Yes, I can.
Okay, good. So I'm Helena Blumen, as Sophia mentioned, and I was trained in cognitive psychology, but I think today I'm more of a cognitive neuroscientist, which means that I try to understand how our brain gives rise to cognitive functions like thinking, perceiving, and remembering. Oops. Sorry, for some reason, I'm not able to move forward. Okay. I am particularly interested in this interrelationship between physical functions like walking and dancing, the brain, and these cognitive functions like thinking, perceiving, and remembering. And what I thought I would do today is to tell you about how we use imagery of walking or imagining yourself going for a walk to understand what brain regions that we use during actual walking. I would like to clarify, however, that imagining yourself going for a walk will not generate the cardiovascular benefits of an actual walk, but as you'll see, the brain regions that we use to complete these two tasks are surprisingly similar. So if my interest lies in cognitive functions like thinking, perceiving, and remembering, why study walking? Well, as it turns out, accelerated gait speed or a slow gait for your age and your sex is a good predictor of both negative physical outcomes as well as cognitive outcomes like cognitive decline, cognitive impairment, Alzheimer's disease, and other forms of dementia. In fact, it's been shown that about 14 years before the onset of mild cognitive impairment, gait speed starts to decline. And this is in men. And in women, it starts to decline about 6 years before the onset of mild cognitive impairment. So clearly, just gait speed is a very good predictor of future cognitive impairment. I think this is because these physical functions like walking and cognitive functions like thinking, perceiving, and remembering rely on shared brain areas or overlapping brain areas. And much of my research so far has been to show that this is the case. And in order to do that, I primarily use magnetic resonance imaging or MRI. So we can use MRI to look at the structure of the brain. We can look at both the size of the whole brain, the size or integrity of particular brain structures. But MRI can also be used to look at how the brain is engaged or how it's activated when we engage in particular tasks like remembering. And this is what we refer to as functional MRI. But how do we use functional MRI to study walking? So as many of you know who has been in an MRI, or I would say even seen one, it is very difficult to walk during MRI scanning. And one way that we get around this problem is to use motor imagery or imagined walking. So motor imagery involves envisioning motor actions without actual execution. And when we use a motor imagery to identify or imagine walking to identify the brain regions that we use during actual walking, we're relying on the assumption that that imagined walking engages the same or similar brain regions as actual walking. And this has actually been shown in the literature. So for example, when we imagine ourselves walking, we activate regions that we know are part of the motor execution and motor planning pathways from the animal literature. So it seems like imagery of walking does indeed engage similar brain regions as actual walking. When I came to Einstein almost 10 years ago now, I became interested in looking at what brain regions that we use during more complex walking tasks, like walking while carrying on a conversation. And in the lab, the way we look at this is by using the walking while talking task, or walking while reciting alternate letters of the alphabet. And this is a task that you as LonGenity participants should be somewhat familiar with. And we use this walking while talking task because it's been shown that poor performance on this task predicts falls, frailty, disability, mortality, as well as dementia. So some years ago now, we developed this imagery of walking protocol to look at the brain regions of this more complex walking performance. So after people had completed the walking and walking while talking task on our mats, we asked them to imagine themselves doing this task first outside of the MRI and then inside or during MRI scanning. And the results of the development and validation of this imagery of walking protocol was published some years ago and also received an award from the American Geriatric Society. And the key finding of this study is shown here in this brain region as red to yellow. So those blobs that go from red to yellow are regions that people activate or engage when they're asked to imagine themselves walking while reciting alternate letters of the alphabet compared to when they're imagining themselves walking or talking alone. And these findings were interesting because these are the regions that we know are important not only for motor functions, but also cognitive functions. So this imagery of walking task and the actual walking task is really a combination of a physical functioning of walking and the cognitive function of reciting alternate letters of the alphabet. So we recently implemented this imagery of walking protocol into the LonGenity study. And this is part of my National Institute on Aging grant that was funded last year. The causal pathway or the hypotheses that we're testing in this study is that functional and structural brain changes actually happen a little bit before we see changes in gait or in cognition. We have already found support for that walking does indeed or walking speed doesn't indeed start to slow before we see changes in cognition. And these are results that we'll be presenting at the Alzheimer's Association's International Conference this upcoming July. Whether it happens in person or virtually, we're not sure yet. We have to collect additional data, both functional and structural brain data, to test the first half of this causal pathway. We are hoping that about 200 of you will be interested in participating in up to 3 MRIs, about 3 to 5 years apart. And so far, many of you, about 115 plus, I would say, at this point, have completed the baseline MRI, so the first MRI. But we're hoping to collect 2 additional MRI for those that are interested. So thank you for those who have already started this process. In this grant, we're also interested in figuring out who might be more vulnerable to these brain changes. And one of the things that we're looking at here is individuals with the motoric cognitive risk syndrome. So individuals with the motoric cognitive risk syndrome tend to walk slower than would be expected of their age and sex. but they also have a subjective cognitive complaint. So they don't have dementia or cognitive impairment, but they have subjectively noticed that they're not remembering as many things as they used to. So they have this complaint about their own cognition. And we have found that that is associated with dementia later on. So we are also trying to figure out who might be more vulnerable or might be less vulnerable to these brain changes. And here we're looking at issues related to social network and social support. So is it the case that people that have socialize with a lot of different people on a regular basis and have the availability of a lot of social support from these people, do they do better? Do these brain changes occur maybe a little bit later in the time course of aging? So those are some of the things that we are looking at in this grant. What we're also ultimately interested in determining is when do we have to intervene. So if these brain changes occur before we see changes in walking performance or cognitive performance, we might have to introduce interventions to prevent or reduce the risk for cognitive impairment and dementia much earlier, maybe going even back to middle age. So we are hoping, or I think we can be quite confident that this grant can provide us a better understanding of this interrelationship between brain aging, physical aging, and cognitive aging. And we hope, as I said, to use that information to develop and test interventions that we can use to prevent or reduce the risk for both cognitive impairment and dementia, as well as mobility disability. So I have very much enjoyed, even though this is over Zoom only, I can't see you in person, enjoyed telling you about how we use imagery of walking to understand what the brain is doing during actual walking and how we can use that information to tease apart the time course of brain aging, physical aging, and cognitive aging. So I want to end with this note that even though we can use imagery of walking to identify brain regions that are important for actual walking, you have to keep in mind that going for a walk is much better or has more cardiovascular benefits than imagining yourself walking. So my advice to you then is that you should keep walking. So that's all I have. I want to thank you again for your participation or your precious volunteering of time to participate in the LonGenity studies. The LonGenity team, my collaborators and mentor, Nir, or Dr. Basilai, Dr. Millman, and Dr. Verghese, the rest of the LonGenity team, Wanda Guzman, who you interact with and who makes all of this happen, as well as my RAs and my postdoc who works more closely with me. So thank you for your time.
Thank you so much, Helena. I think we all learned a lot. And thank you for highlighting the importance of still going out for a walk. And we have a lot of questions coming in, which we will save for the end. And now I will present Dr. Sandra Alexic. Sandra is a physician trained in internal medicine and endocrinology who completed her fellowship at Einstein and then has done additional training in metabolism. And since completing her clinical fellowship, she has really married her interest in metabolism and aging, where she's now studying how brain regulates aging, and specifically through some metabolic and hormonal pathways. Sandra received a Resnick Emerging Scholar in Aging Award from Einstein, and recently has been awarded an Outstanding Abstract Award from the Endocrine Society for the work that she will share with you today. Sandra.
Thank you. So I will share my slides. Okay, so can you all see my slides? Yes. Okay. Thank you so much for this kind introduction and for the invitation. It is a true pleasure to be here today virtually with all of you and to address a very intriguing question. Do men experience menopause? As you probably know, the world is aging. Between now and 2050, the number of people who are age 80 and older will triple, comprising about 5% of the global population and 7% of the population in the United States. However, as a society, we are not entirely prepared for this drastic demographic shift that's about to occur. And one of the many reasons for this is that we do not quite understand the changes that occur in human body at the extremes of old age. Indeed, in order to be better prepared for these changes that are about to come, we must improve our understanding of the normal physiology in people with longevity. And thanks to you and other people like you who participate in our study and other similar studies, we are now, for the first time, we have the opportunity to actually learn more about the functioning of the human body at the extremes of old age and help bridge this knowledge gap in order to meet these changes more prepared. Among organ systems that change most drastically with aging is endocrine system, and endocrine function in general declines with age. This is true for growth hormone, this is true for thyroid hormones, and this is also true for sex hormones. And when we think about sex hormones changes with age, usually the most obvious one is the menopause. Menopause is a quite abrupt cessation of reproductive function in women, which inevitably occurs. In the US, the average age is about 50. However, what we still don't know is if men also experience menopause. So we decided to study this question first in our Longevity Genes Project cohort. As Sophia mentioned, many of you are familiar with this cohort that included centenarians, their offspring, as well as the control subjects who did not have centenarian parents. Many of you had parents who were in this study, or you yourselves were part of this project. So we included 427 men from Longevity Genes Project cohort, age range 50 to 106 years. Among these men, around 20% or 84 men were age 90 and older. So what did we find? So graph that I'm showing here is representing the levels of total testosterone by age. On the x-axis, we see age groups that start from 50 and end at 100 and older. And on the y-axis, we see total testosterone levels. So what we can appreciate here is that men tend to maintain quite normal testosterone levels up until age of close to 90. And then after the age of 90, the levels of testosterone seem to decline quite precipitously. Now, this is suggestive of menopause, but it still does not confirm that what we are seeing is truly a male menopause. So in order to address this question in a little bit more depth, let's first step back and take a look at normal regulation of reproductive endocrine systems and menopause itself. So it all starts at the level of the brain. The main master regulator of the endocrine system of our body is called hypothalamus. Hypothalamus, here depicted with this blue light, is a small part of the brain which is positioned roughly behind our eyes, and it is closely connected with the pituitary. Pituitary is a small gland at the base of the brain. Together, hypothalamus and pituitary direct the endocrine function of the whole body. When it comes to the female reproductive system, hypothalamus signals to the pituitary to make its own hormones, and then pituitary makes hormones that stimulate ovaries to work. Specifically, one of the hormones from the pituitary that we are interested in is luteinizing hormone, or LH, which tells the ovaries to produce their own hormones such as estrogen. This whole system is regulated like a negative feedback loop. And maybe the best way to explain this loop is by thinking of a thermostat. Let's say you have a room with a room heater, and that heater is connected to a thermostat, and the thermostat is set at 70 degrees. So if the temperature in the room is below 70 degrees, the thermostat will keep sending the signals to this heater to keep heating the room. However, once the temperature in the room reaches the set point of the thermostat of 70 degrees, thermostat will stop sending these signals. So it works quite similar with endocrine system. Specifically on the example of the female reproductive system, what happens is that as estrogen levels go up, this signals back to the hypothalamus and pituitary to reduce the production of LH. On the other hand, if estrogen levels go down, hypothalamic thermostat will sense this, and it will send the signal to increase production of LH and ultimately to increase production of the estrogen. So what happens in the menopause? Well, in the menopause, essentially ovary runs out of eggs and develops failure. So the primary problem in the menopause is the ovarian failure. This will cause estrogen levels to go down. If estrogen levels are low and the whole axis is functional, then the hypothalamus will sense this. And it will send the signals to the pituitary that will then send signals to the ovary to try to make ovary to work harder. So essentially what we are measuring in the blood is low level of estrogen and high level of LH. And this is how we know that a woman is in menopause. What happens in men? The whole system is regulated in quite a similar way. So hypothalamus and pituitary send signals to testes through hormone LH, which then signals to testes to produce testosterone. Testosterone again feeds back in that same thermostat-like negative feedback loop to downregulate its own production. So if testosterone levels go down, the hypothalamic thermostat will sense this, and it will send signal to the pituitary to increase the level of LH. And in that case, we should measure high LH level. So what did we find so far? Well, we found that testosterone levels go down. It seems that they go down quite abruptly after the age of 90. However, if we really think that the problem is at the level of the testes, which would be similar to what we are seeing with ovarian failure in female menopause, then we also need to expect that LH levels will go up. And indeed, the next step in our analysis was to check what's happening with LH levels. So when we analyzed LH levels in our participants, which are presented on this graph, we found that around the same time where testosterone drops, LH starts to go up quite abruptly. And again, on this graph, we can see that this happens around age of 90. If we plot testosterone and LH together on one graph, with testosterone being a blue line and LH being a red line, we can see that they pretty much mirror each other. Around age of 90, when testosterone starts to drop, LH starts to rise more abruptly. And taken together, this kind of hormonal picture corresponds with what we are seeing in the female menopause. So far, it does seem that men do develop male menopause. But the next question was, how often does this happen? We know that in women, all women get menopause eventually if they live to the age of menopause. But do all men get menopause? So what we've found in our subjects is that if we look at the men who are younger, which are presented here in blue bars, those are men who are in their 60s and their 70s, we really see very few % of men who have male menopause. As men enter their 80s, the percentage of men with male menopause increases progressively, and it keeps increasing. And once they reach their hundreds, which is the group that's presented with the lightest gray bar all the way to the right here, we see that more than 50% of men develop male menopause. So taken together, it seems that men do develop menopause, and maybe not all of them develop menopause, but quite a substantial proportion do. So next question was, well, is male menopause harmful? And this is a very important question because the studies that have been done in a general population of aging men, meaning men who do not necessarily have a predisposition for longevity, have found that low testosterone may be associated with all sorts of negative outcomes, including poor metabolic health and worse survival. Nonetheless, these studies did not include men with longevity, and also they did not look specifically at men whose hormone levels were changed because of the male menopause. So we wanted to know, in men with longevity specifically, is male menopause associated with negative health outcomes and worse survival? So I mentioned that one of the health outcomes that's been associated with low testosterone is poor metabolic health. So this is what we wanted to investigate as well. Briefly, what we found is that in men with longevity, low testosterone was not associated with poor metabolic health. Now I'm going to walk you through this in a little bit more detail by looking at these specific graphs. So on these graphs, we are looking at men with longevity, with blue bars representing men who had normal testosterone levels, and gray bars representing men who have low testosterone levels. If you look at your left upper corner, we're looking at the BMI. BMI, or body mass index, is a measure of obesity. What we see here is that men with normal testosterone compared to those with low testosterone had a similar presence of obesity. And then if we go to the graph to the right of this one, which is showing blood glucose levels, we see a very similar situation. The men with low testosterone and men with normal testosterone have similar blood glucose or blood sugar levels. Then to the right, in the right upper corner, there are triglycerides, which are type of the fat or lipid in the blood. Again, very similar levels in men with low and normal testosterone. And the three graphs on the bottom are showing total cholesterol on the left, then your bad cholesterol or LDL cholesterol in the middle, and HDL cholesterol or good cholesterol on the right. In all three cases, there were no significant differences between men with normal testosterone and low testosterone. So from our data, it appears that low testosterone in men with longevity is not associated with poor metabolic health. But is it associated with worse survival? So we also wanted to look at that. Let me just guide you through this graph briefly. On the x-axis, we are looking at survival age. So essentially, this is the age where our participants died, each of them. And on the y-axis, we are looking at the proportion of men who were alive at any of those ages. And we can see that at the very beginning, the men, all of them are alive, so proportion is 1. And then as time goes by, these proportions decline. Now, men with low testosterone are presented with the gray line, and men with normal testosterone are presented with blue line. And what we can see just by looking at these lines is that they pretty much overlap. It does not really seem that they are different. And actually, when we ran our statistical tests, we confirmed that there was no difference in survival between men who had low testosterone and those men who had normal testosterone. So in conclusion, we found that men may experience menopause, which happens not in all of them, but in quite a high proportion of them. And it occurs about 40 years later than in women. Male menopause was not associated with the negative health outcomes, at least the ones that we studied. So this poses a question that has a very significant clinical implication, which is, should testosterone replacement then be offered to men who experience male menopause? And our data suggests that maybe this is not needed. Nonetheless, the data that I presented comes from cross-sectional study. This data is very valuable, but it's extremely important to confirm our findings and to further expand our findings in the longitudinal study. For this, we're planning to use the data from LonGenity, which allows us to actually track levels of hormones in each individual participant over time. And again, thanks to all of you who are participating in LonGenity, we will be able to look into this question in even more detail and to confirm and expand our findings. So we are planning to confirm that men may experience menopause, but that it is not associated with negative effects on metabolic health or survival. Then, thanks to the plethora of data that's been collected in LonGenity, we can also look at the other health outcomes different than the ones that we already studied. And finally, through our LonGenity MRI protocol that some of you are already part of, and we are really thankful for that, we are planning to explore if the structure of the hypothalamus has impact on its functioning as a thermostat, and also if it's related to health outcomes. In the end, I would like to thank with all my heart to all of you, our study participants and your families who are making this possible and who are working with us to extend the limits of our knowledge. I would also like to thank our longevity research teams, my mentors, Dr. Sophia Millman and Dr. Nir Barzilai, our collaborators and funding agencies, and we'll be happy to take your questions. Thank you.
Thank you so much, Sandra, Helena, and Nir for your presentations this afternoon. And we will now open the floor to questions. So I will begin by asking the first question. This question is for Nir. Nir, what factors determine how we age?
What I brought here is I brought the concept of hallmarks of aging. Okay, those are the things that are happening with aging, and that if we can fix them, they can fix aging. Okay, that's our scientific answer for that. And to make a bigger answer, I will have to go into details, which either you can see in the book or call me later and I'll talk with you. But the point is that we have a pretty good grasp, at least initially, a good grasp of what's leading the aging and how we can target it and delay it and stop it and reverse it in several ways.
So just a follow-up to that question, do you think it's all in the genes, or does our lifestyle make an impact?
Okay, that's a really good question and very appropriate here. Look, there's no doubt that if you can do something now without waiting to drugs, is to move more, to exercise. Exercise is great at any age, any sex, any place in the world. Exercise is number one. Really doubling on Helena's final words, walk, move, and not in MRI. The second thing that's important is diet. Now, this is the interesting story from my lab. You know, we always use caloric restriction. If we take mice or rats and give them less food, they live significantly longer. And this was the basic of all the biology of aging. And we took it to say that if you have less for breakfast, lunch, or dinner, then you're set to age slower. But in fact, what we've done is we gave all the food in the morning to those animals, and they were hungry, and they finished all the food in 20 minutes, and then they were fasting for 23 more hours. When we start giving them the food throughout the day, they were thinner because they had less food, but they didn't live longer. And now our concept that's relevant to aging is to do prolonged fasting. I myself am doing 16-hour fast every day. I'm basically skipping breakfast, but not only breakfast, no bite, no cheating, for 8, you know, I count. If I stop at 8 o'clock to eat, I won't start between noon times to replicate this idea of fasting. So I'll stop here, but yes, exercise and nutrients are the main thing. But for centenarians, it's more genetic. And that's what we want. We want the genetics so that we can stop the bad effect of the environment by designing drugs that will help do that.
Thanks, Nir. So to follow up on that topic of lifestyle, this is a question for Dr. Blumen. Can gait training or exercise affect your cognitive function?
So there is some evidence for that gait improves walking, or gait training improves gait ability and gait speed. And I would say the evidence is modest in terms of improving cognitive function. So there's clearly evidence for it, but it's not as convincing as we would like it to be. And I say that partly because many of the studies that we do, or the trials that we run, have a limited amount of time. So they're either 3 or 6 months long. So we do see some modest changes, but not as much as we would like to see. And that's probably because we don't follow people for long enough time to see these more convincing effects. But so there is some evidence, although it's quite modest.
And do you think that imagining walking will actually improve your actual walking?
I hope so, because I have a clinical trial that does look at that issue. I do have a clinical trial in older adults to look at that. So we do imagery training to see if it's effective in healthy elderly adults. It has been shown to improve recovery after a stroke, for example. So if you have a stroke and you lose the ability to walk, or walk slower than you would, if you combine rehabilitation with imagery training, you recover faster than if rehabilitation alone. And it's also been shown to improve gait or walking in people with Parkinson's disease. But we're hoping that it can also be used to improve walking abilities in healthy elderly or normal elderly adults.
Thank you. And the next question is for Dr. Aleksic. Do you think there's a role for testosterone replacement in men, in older men? We know there have been studies in older women, primarily the Women's Health Initiative. Have there been similar studies in men? And would you recommend giving testosterone to older men, maybe who are not centenarians?
Well, I think that's a great question. It's a question that's actually still open, and I don't think that even the endocrine community has a clear answer to that question. So as you mentioned, Women's Health Initiative was a really, really big study that involved women who were postmenopausal and were treated with hormone replacement with the goal and hope that this will actually improve their health and improve their survival. However, it did not really work out all that well, and we do know much about risks of hormone replacement therapy in women thanks to that study. When it comes to men, we do not have really a study that was of the caliber of Women's Health Initiative study. There have been clinical trials looking at replacing testosterone in older men, but these studies were generally shorter and smaller. It gave us some insights. They gave us some insights, but not clear answers to all the questions. So we know, for instance, if testosterone is given to older men, we know that this will improve their sexual function. We know that this will improve their red blood cell counts, meaning that if they have anemia, this will get improved. But then for many other outcomes, we either have not found benefits, like no clear benefits, for instance, for cognitive function, no clear benefits for physical function. And also when it comes to the outcomes that really worry about like mortality, cardiovascular events, we really do not have good data to know whether this is helpful or could even be harmful. One of the problems is that people did not really look in these studies why men have low testosterone, meaning if this is something that's similar to what we showed in our participants, which would be very similar to menopause, where your testes are kind of failing to produce enough testosterone, but your brain is working totally fine and responding properly to that. Or it might be a different reason where your testosterone is low just because you have unhealthy aging and you're not making enough testosterone because of that. So I would say that we are still not sure about this. There might be some benefits, but the risks are still unknown, and especially long-term risks, we need to learn more about.
Thank you. So the next question is for Dr. Barzilai. Nir, can you share with us some of the genetic discoveries that have been made to date, some of the genes that may confer longevity or protection from Alzheimer's disease?
Sure. What's really interesting is this story that in nature, the dwarfs are living much longer. Like that the small dogs are living longer, and the ponies are living longer than the thoroughbreds. And we can go on and on, from mice who were born dwarf and flies and nematodes. And it was weird for us to think and anticipate. I actually had a grant where I said that's happening in human, but I didn't believe that at all, only to prove that it's the opposite. And Sophia, you might talk about the phenotype, but basically 60% of our centenarians have some alteration in genes that decrease growth. And the reason for that is probably that we need growth in order to get to reproduction. And, the bigger the men are, it's better for them to fight and stay alive, et cetera, et cetera. We need growth, it's good. But when we age, we need to shift the energy. We now need to stop the breakdown, okay? And we cannot spend energy on growing anymore. And so it happens that those who were born with some alteration in the growth, and some of them are like shorter, okay? But they benefit in aging, and they have lower growth hormone action and less disease and better health span.
Thanks. And to follow up on that, do you think these genetic changes are specific to Ashkenazi Jews? And more broadly, why are we focusing on Ashkenazi Jews?
Well, that's a good question. And that's part of the informed consent explanation that you sign. And the reason is our history, okay? Our history that is not such a great history resulted on us, after years of isolation and persecution and death and everything, on being relatively homogeneous genetically. We are more alike each other than the rest of the white people. And from a genetic point of view, it means that we need like 50 times more other white people in order to get to the same results. And indeed, although we have just hundreds and not thousands people, we still get quite incredible results that can be also validated then in other population. There's another reason to choose the Ashkenazi that is really very important in the United States. There's a health disparity according to education or to socioeconomical factors in the United States. The more uneducated and poor you are, the less you get to age. You'll die sooner. And so this homogeneity is very important, because your average education is 17 years, right, Sophia? Something like that. I mean, you're educated, which means you're not homeless, you're all taken care of. So we take this away as a diverse point for us. So the homogeneity, both genetically And both socioeconomy is really important. And it's not because we have any indication that Jews live longer or shorter.
Thanks. This is a question that's open to the group. What do you see as kind of the next developments in the treatment of aging? Where do we see this field going? And what do you kind of see on the horizon in terms of interventions that we can use to delay aging?
If none of you want, I'll be happy, because it's picking back on another question that I saw of somebody hearing David Sinclair's talk, and he said he's taking NMN. Should we take NMN? Okay, and I want to make one point. We have many drugs that can be repurposed for aging, but what we need is clinical studies, okay? Not observation and not studies in animals, but clinical studies. What's a clinical study? You take a population, like was mentioned with the Women Health Initiative or with the testosterone studies. Half of them get the treatment and half of them do not get the treatment. And somebody independent is there to watch and independent to analyze the data. And only if the data support the hypothesis, I can say, you know what? I'm confident that that's the drug you can take. Okay, this example of the NMN, which is a kind of a nutrient that you can get over the counter, and which I've tried and I didn't see any effect that I wanted to. It's hard to see effect that stops aging, okay? But the point is that it's very promising. David Sinclair does it in mice, not in humans, okay? It's very promising. It looks like it could be translated. It looks like it's safe, and that's why I was trying that. But there's no evidence that it's doing anything in humans. And I'm spending time in my book talking about it because we all should do better. When we want to take something, we have to look for the clinical data and not just read a headline of something or read an advertisement of somebody said, hey, I have something you'd live forever. That's not how it's going to work for us.
Well, I can answer my view of where the field is going is I'm not as much an expert on pharmacological intervention, but in terms of lifestyle intervention, I think the field is going more towards multimodal intervention. So maybe not necessarily, well, we know aerobic exercise might be efficient, but maybe combining aerobic exercise with cognitive training or with social interaction. So one of the studies, for example, that we just completed looked at social dancing, which is kind of a combination of both social interaction, cognitive training, because you have to remember where and what to do in terms of movement, but also gives you a little bit of a aerobic exercise.
I can chime in as well. So, you know, kind of piggyback on what Nir said about having clinical data and having data in humans. Where I see the field is going in terms of at least pharmacotherapy is repurposing. I think that what we will be seeing in the next years will be clinical trials that will be looking at the drugs that have already been used in humans and have some beneficial effect that at least suggest a modifying of aging process, and then repurposing these drugs for aging specific indications. So I think that that will be the first step maybe that will occur before translating the drugs that have already been used only in animals to humans. So I think that repurposing will be the next step.
Thanks. And I think something that's still on our minds a lot is COVID, right? Because it is still so much with us. And so a question came in, you know, if we know that social interactions are good for us, do we think the social isolation that many of the older adults have experienced over the past year could or would have a negative impact on their cognitive function? Address it to Helena.
I think so. I think there might be some negative effects of not being able to physically interact with the people that you normally do because of social distancing requirements of this pandemic. But I also think that we might find people that do better. So I think it's possible that because of social distancing, you become a little bit closer to the people, the very close people, meaning your parents, your children, your grandparents, that you might not necessarily be able to see them, but you might actually check in with them much more often because of this pandemic. So I think we're going to see both things. We're going to see people be negatively affected by this social distancing, but also we might discover people that are a little bit more resilient because now, because of the pandemic, they're being more careful about calling up their grandparents and making sure that they're okay, which might not have been the case when they had to go to work every day and had so many other things that they were working in. So, but I think time will tell. But those are the two scenarios that I see.
Can I add something? I think that this period was generally cruel, very cruel to the elderly, in particular those that were in old age home. It was really cruel. They were lonely. And I'll just tell you an anecdote from our study. We had a centenarian who was 106-year-old, and she was doing relatively well until this happened. And then she lost all social contact to the extent that she stopped eating and drinking and died within a few days. The COVID virus didn't kill her, but the COVID period killed her. And this is, I think, heartbreaking, and that's what's much on my mind.
Yeah, I think we all echo those sentiments. So here's a different question, and that is, why do we keep asking the same questions? Why do we keep giving the same tests year after year to our participants when many of them, it seems like, have aced them? Why do we have to keep giving the same tests and we can't change it up a bit?
Sophia, I think you're the best to answer that.
Well, I would like to give the floor to maybe our psychology colleague, since a lot of these tests are neuropsychological. Helena, you want to take it? And then I can chime in.
Sure, I can take parts of it and anybody can chime in. So because these tests that we use have been normed and validated in many different populations, when there is a change, even if you have repeated this over time 10 times, if we do see a change during the 11th time, we know that that is meaningful, and we know that that might indicate that something is wrong, that there's something that we have to do. So that's why we have to, unfortunately, repeat many of these tests over and over again. And we can actually look at how it changes, so across time, and which functions. Is it memory that declines faster? Is it attention? Is it all sorts of different aspects of cognition? So repetition, unfortunately, is very valuable to us because if we do see a change, we know that it has meaning, and we can compare it with other studies as well.
Can I give an example? I want to give the example of Jerry Rubinstein, who's actually listening now. He asked one of the questions, and he says there, I'm 91 years old and I'm there from the beginning. Basically, it's been 18 years. And Jerry is really, he's my father-in-law, by the way, and he's incredible. And I would say that even if he wasn't listening. And And Jerry is one of those guys that one of the questions that you have is count in 1 minute as many as fruits as vegetables as you remember, okay? And in 18 years, every time that he comes, he gets more because he knows the question is coming. And the day before, he goes to the market and he sits there and looks at the vegetables and remember them also visually from left to right. And he often has fruits that our testers had no idea, never heard of. So the point is that a lot of you guys have better scores as we go on. And it's always a problem for us to present it in the right way so that people don't ask us, how come your guys, when they get older, they get better cognition?
Right, and for those who may not be satisfied with answering the same questions over and over, we do try to implement new cutting edge science research, which includes the MRIs and the CAT scans and additional questionnaires focused on social networks, and now the COVID component. So we are certainly trying to, I would say, make the most of the study and make the most of your contributions and stretch it a little longer to answer more and more scientific questions that we can about aging so that we can all benefit. So in the final minute, I will just pose a question to all the panelists today, and thank you again for participating. And that is, what do you look forward to in the future, and what do you think would be kind of the next revolution in aging? Can start with Nir.
Well, I'm very optimistic. There is a new foundation that's going to spend $1 billion a year in stopping aging. And this is the kind of resources that we needed, because we're kind of in silos. The National Institutes of Health, they're actually Institute for Disease. The only one who cares about aging is the National Institute of Aging, and it doesn't have enough budget because of the other silos. So I think we're going to have enough money to take what Sandra said before, drugs that we know that if we repurpose are going to have potentially going to extend health span and lifespan, and we can start doing it immediately. And in the meantime, developing pipeline of better drugs, combination of drugs, new drugs that can really, really stop aging and allow us to use those years much better and healthier and happier than we did so far.
Sandra?
Can I be next? Yeah. So so I think that I agree absolutely with Nir that with more money coming in, there will be more people coming into the field, and what I particularly look forward to is more people from different disciplines entering the field. Just like from my own experience, I entered the world of endocrinology. I always thought that I was going to be an endocrinologist, and then I discovered aging, and it was a mind-blowing revelation. So I really look forward to more people, more scientists from different disciplines entering the field and working together to really move this forward. I think that the fact that people are actually recognizing that we need to modify aging process has been an enormous progress already. And I look forward to the field growing.
So just to add on to Sandra's point there, I think in addition to different disciplines, maybe the collaboration between clinicians and basic scientists like myself, that I'm looking forward to as well.
And I think that's a really nice note to end on, because I think aging is probably very ripe for that. We have clinical scientists and basic scientists very closely working together, and especially in our group. I think our group, our study of longevity and our aging studies, our longevity studies here at Einstein really exemplify that kind of collaboration between basic scientists and clinical scientists, and taking what we learn from each other and cross-testing it, so to speak, right? Bringing the basic science into clinical work and vice versa. So with that, again, I want to thank our panelists today, Dr. Alexic, Blumen, and Dr. Barzilai. And most importantly, I want to thank all of our LonGenity participants and our Longevity Genes Project participants and our lifelong participants who have really made this research possible and are really forging the way for the future. If it weren't for you, we wouldn't be able to make these discoveries and bring, the bright new future into reality. I want to let you know that we are in the process of reopening the LonGenity study visits. Like everybody else, we were on hold and working primarily remote for quite a bit of time. But as things are looking much better and most of our study staff have been vaccinated, and I know many of the participants probably have as well, we are looking to resume our usual activities in our research centers at Albert Einstein. So you will be hearing from us soon. We're starting out slowly, taking it one day at a time, but I would imagine that by fall, assuming that things continue to look up and look well, we will be back in full swing. So we do look forward to seeing you in person soon. And until then, please reach out with any questions, comments. We'd love to hear from you. Stay safe and stay healthy.
Thank you.